Thank you very much for this invitation. It gives me the chance to try to convey some ideas and also to give an update of what we know about the mechanisms of action behind the psychostimulant effects of caffeine. First concept is that caffeine is a psychostimulant with the same central effects as classical psychostimulants, as cocaine and amphetamine. So it produces increase in motor activity, has arousal effects, and has reinforcing effects. Even though it nevertheless can produce some dependence, let's say also that it doesn't have this very strong reinforcing effects than cocaine and amphetamine. It is well known that the mechanism behind the psychostimulant effects of cocaine and amphetamine are due to an increase in their ability to produce a stimulation of the dopaminergic system, particularly in the striatum. I'm talking about the motor effects and reinforcing effects. And the striatum is this area, is a structure. It's called the input structure of the basal ganglia. And these are involved in the elicitation and learning of reward-related behaviors. And this area contains the highest concentration of dopamine and also the densest concentration of dopamine receptors in the brain. And the ability of cocaine and amphetamine to produce those psychostimulant effects is by the ability to bind to what is called the dopamine transporter. And by doing so, either because what cocaine does is antagonizes, blocks its effects, or what amphetamine does is reversing its effect, what it produces an increase in the extracellular concentrations of dopamine. That's a presynaptic effect, which turns into a stimulation of postsynaptic dopamine D1 or D2 receptors that we will see later. They're localized in different neuronal components in the striatum. It's been known, well, at the beginning of the 90s, we already knew that the main mechanism responsible involved in and the central effects of caffeine were adenosine receptor antagonism. And we know there are 4 adenosine receptors, A1, A2A, A2B, and A3. And it was known that the concentration reached by just taking coffee, that would be enough to block the effects of endogenous adenosine on adenosine A1 and A2 receptors. Adenosine A2B, they have really low affinity for adenosine, so they are not involved unless in pathological situations. And A3, they have very little affinity for caffeine. So the question was how adenosine modulates the dopaminergic system to modulate motor activity and reinforcing effects. Then also at the beginning of the 90s, then we already knew that caffeine didn't have a very clear, strong presynaptic effect. It did not really increase dopamine in the extracellular space in the brain. And then we thought that there should be a postsynaptic mechanism, some kind of interaction between adenosine and dopamine receptors at the postsynaptic side. And we used this reserpinized mice model to test that hypothesis. So what reserpine does, what you administer systemically, is that depletes catecholamines from the brain, and then the animal becomes immobile, cataleptic, and then the only way to counteract this effect, to produce motor activity, is to give a dopaminergic agonist. For instance, bromocriptine, that is a dopaminergic D2 agonist, as you can see, produces locomotion in these reserpinized mice. And then what we saw is that was dose-dependently inhibited by adenosine receptor agonists with a potency that was suggesting that adenosine A2A receptors were involved. At that time, we didn't have really selective adenosine A2A antagonists. So you can see the reduction by these NECA and LPA adenosine receptor agonists. And on the other hand, caffeine or other xanthines, like the metabolites of caffeine, theophylline, and paraxanthine, but not theobromine, which is, by the way, very low potent adenosine antagonist, and then they would do the opposite effect. They would not have an effect on their own. They would not counteract the immobility of reserpine, but they would potentiate the effect of bromocriptine. So this was showing us the existence, probably we were postulating the existence of some antagonistic interaction between adenosine, A2A probably receptors, and dopamine D2 receptors. And that would suggest that adenosine A2A agonists would be like dopamine D2 antagonists. and that adenosine A2A antagonists would act as dopamine D2 agonists. So we show for the first time that an A2A receptor agonist would produce catalepsy, as a dopamine D2 receptor antagonist would do. And we also showed later on, when we had availability of adenosine A2A receptor antagonists, selective adenosine A2A receptor antagonists, that those compounds, sorry about these graphs, just to show the different selective A2A receptor antagonists available in the market, then would produce motor activation, the opposite effect. So this data suggested that caffeine would produce motor activation, or that would be a mechanism involved in motor activation induced by caffeine, the ability of caffeine to block adenosine A2A receptor mediated inhibition of dopamine D2 receptor activation. Then we found evidence for a more direct interaction, really direct interaction between the two receptors, A2 and D2. And this, sorry about this graph, is just to show that in very artificial preparations, membrane preparations of rat striatum, the dopamine D2 receptor antagonist could be displaced dose-dependently by dopamine. Nothing new about that. But what we showed is that this displacement, the ability of dopamine, the agonist, to displace the antagonist, was modified by the addition of adenosine, by an adenosine A2A receptor agonist, the CGS 21680. And when we analyze the parameters of these competition curves, it shows us that it was due to the ability, so the A2A receptor agonist would decrease the affinity of dopamine D2 receptors for dopamine. That experiment demonstrated that those receptors should be colocalized in the same neurons, and they should interact in the same neurons. But which neuron? And this neuron is called the efferent striatal GABAergic medium spiny neuron, MSN for short. If we go back to the striatum, this neuron constitutes more than 95% of the striatal neuronal population. And this neuron receives afferents, and I keep asking, sorry for that, from glutamatergic inputs from cortical, limbic, and thalamic areas, and the mesencephalic dopaminergic input from the substantia nigra and the ventral tegmental area, the mesencephalon. And then we have two subtypes of GABAergic striata of MSNs. These neurons, they give rise to the two main efferent pathways from the striatum, which connect the input structure of the basal ganglia, which is the striatum, to the output structures of the basal ganglia, which are the substantia nigra pars reticulata and the medial segment of the globus pallidus. The called direct MSN, which connects directly the striatum with the output structures. This neuron contains dopamine receptors of the D1 subtype and contains certain peptides. And the other neuron connects the striatum with the lateral segment of the globus pallidus. This is the neuron that contains dopamine D2 receptors. Well, we know now that this neuron also expresses adenosine A2 receptors. So we look at different preparations of mRNA, of A2A receptor mRNA, or D2 receptor or A2A receptor protein in different species. We see that adenosine A2A receptor in the brain is really concentrated in this structure, which is the striatum. And what you're looking at here, by the way, in fact, is this indirect MSN, the neuron that contains also the dopamine D2 receptors. Then we found a neuronal correlate of this adenosine A2 dopamine D2 antagonistic interaction. And this is microdialysis experiments in rats, in awake animals. What we do is we implant one probe in the striatum, we deliver certain drugs, and another probe in the localization of the terminals of this indirect MSN, which is the globus pallidus, and we measure GABA levels. Then we stimulate with dopamine agonists, different compounds, the cell bodies, and we study the release of GABA. What we could see is that we would give dopamine D2 agonists in the striatum, we would have a decrease in the levels of GABA that will be counteracted by the coperfusion of adenosine A2A agonists. And that would be the opposite, would be potentiated by the perfusion of theophylline in that case. So that will be a correlation at the neural level of the antagonistic A2 dopamine D2 interaction. Then I need to tell you about two concepts that we are now using in pharmacology that are really important to understand the effects of caffeine at the central level and many other compounds. The first concept is going to be receptor heteromers, and the second is the local module. So since 1878, the receptor concept, it's been the main concept when it comes to receptors, and they will be considered receptors as a single unit. But now we are changing our way of thinking by including the term receptor oligomer. It can be receptor heteromer or homomer. So receptors are not only just one receptor, but they are associated receptors. They're included in macromolecular complex made of different receptors. And that's the way we define receptor heteromer, which is a macromolecular complex composed of at least two functional receptor units with biochemical properties that are demonstrably different from those of its individual components. The second concept, local model, we go back to that neuron, the MSN. I was telling you that this neuron receives two main inputs, corticolimbic inputs, glutamatergic inputs, and mesencephalic inputs. These two inputs converge into dendritic spines of these neurons in a way that the glutamatergic input makes synaptic contact with the dendritic spine, with the head of the dendritic spine, while the dopaminergic input makes synaptic contact with the neck of the dendritic spine. So these elements in the dendritic spine, the glutamatergic terminal and dopaminergic terminal, and then glial processes that wrap around the glutamatergic synapse constitute what we call the striatal spine model, which is an example of a more general term that we call local model, that we define as the minimal portion of one or more neurons and one or more glial cells that operates as an independent integrative unit. Why am I telling you all this? Because this is the frame that we need to understand what it's called extrasynaptic transmission, not only synaptic, but extrasynaptic transmission. I'll tell you what it is about. And also receptors that are extrasynaptically located, not only in the synapse. And also, we can then understand what receptor heteromers are and what's the functional significance of them. So with this cartoon, I tried to convey that effect. So dopamine, which is in red here, the dopaminergic terminals, is not only released synaptically, but also extrasynaptically diffuses and also stimulates dopaminergic receptors, which are located in these different elements of the local module. So not only pre- and postsynaptically at the dopaminergic synapse, but also pre- and postsynaptically at the glutamatergic synapse. The same for glutamate. Glutamine is not only released intrasynaptically, it also spills over and stimulates glutamatergic receptors, which are localized in different elements of this. So look at the complexity of all this model. Then, which is what, in fact, is really closer to the truth than just strictly synaptic transmission. And then adenosine is coming in form of ATP released from the glial processes and converted to adenosine immediately by 5 nucleotidases. And again, this adenosine, it travels in this local module to stimulate adenosine receptors, which are located everywhere. Okay, the adenosine A2 and dopamine D2 receptor I was telling you about, they are located right there, perisynaptically, not even intrasynaptically. Then, sorry about that, it's just to tell you that we have done a lot of research using artificial systems, have done a lot of research using biophysical methods called BRED and FRED. They are resonance energy transfer techniques. And also we use mass spectrometry using also peptide-peptide interaction studies to demonstrate that in these artificial, in transfected cells, we can get A2 and D2 receptors get together. They form a unit. That's where caffeine is going to act, blocking the A2 receptors on the A2D2 receptor heteromer. We also have found intimate intermolecular interactions. We have found epitopes of the two receptors that they bind together. And that includes an arginine-rich domain in the third intercellular loop of the D2 receptor, and then a portion of the C-terminus that is of the A2 receptor, where they get together by electrostatic, very strong electrostatic interactions. They're even covalent-like in terms of strength. Why I'm telling you this? Because we reached also the understanding of this interaction at the cellular level. So in patch clamp experiments, in identified neurons, you can identify neurons that express D2 receptors because you use animals, transgenic animals that express D2 receptors in cells that you can see with fluorescence. They have GFP in those cells, so you know exactly which cell you're patching. So in these cells, you can see that the activity of the cells this is the firing rate induced by NMDA, by the application of NMDA, can be counteracted by a dopamine D2 receptor agonist. This is MPA, propylapomorphine. What I want you to see is that this effect, the effect of the D2 on inhibiting the activity of this neuron, is completely counteracted by CGS. And what is wonderful about that I couldn't help to show you, is that if you now infuse peptides that are interrupting that electrostatic interaction, you interrupt this pharmacological and this functional interaction. So we really have reached the level of understanding of at least one mechanism of action, which is the adenosine A2 dopamine D2 receptor heteromer in this particular neuron. So this A2-D2 antagonistic interaction mediated by A2-D2 receptor heteromers can explain the depressant effect of adenosine A2 receptor agonist I told you before, and the motor activating effect of A2A antagonists, including also at least the A2A receptor mediated motor activating effects of caffeine. This is so important, and caffeine has been so important in this story that led us to the speculation at some point that we could use A2A receptor antagonists in Parkinson's disease based on this interaction. And it's being used as we speak. So there are many clinical trials now, and some that have been already positive, showing that A2A receptor antagonists potentiate or improve the therapeutic index of L-DOPA based on the A2-D2 antagonistic interaction. But you will see caffeine is a really incredible substance. It has so many other effects, and not only mediated by adenosine, but mostly the ones mediated by adenosine. I said at the beginning, some were A2A, and others are A1 mediated, right? So the fact is that if we give an A2A receptor, that would be the dose response induced by caffeine, the motor activity, locomotor activity in rats induced by caffeine. That would be the one produced by an A2A receptor antagonist, and that would be the one produced by, in blue, produced by an adenosine A1 receptor antagonist. So A1 receptor antagonists also produce motor activation. And to make a long story short, and also take my word for it, using all these methods and approaches I've been telling you, we've been able to show that also there is an antagonistic A1-D1 receptor interaction in the other neuron that I was telling you about, the direct MSN, that it's also mediating part of these effects. So that would be another postsynaptic mechanism. That would be the one I told you before, A2-D2 in the indirect MSN, also called encephalinergic neuron, and that would be the A1-D1 receptors forming heteromers there that would also mediate these postsynaptic effects, A1-mediated postsynaptic effects of caffeine. We did also microdialysis experiments showing that that happens also at the neuronal level. And finally, I'm almost done, there are not only postsynaptic mechanisms, we also found presynaptic mechanisms after all. I told you that there was no evidence that caffeine could release dopamine like amphetamine or cocaine. Well, it does in a particular part of the basal ganglia of the striatum, in the most ventral part of the striatum, which is the shell of, is called the shell of the nucleus accumbens. We found that caffeine releases dopamine and releases glutamine in this structure, but it's a less potent effect than amphetamine. That's due to adenosine A1 receptors localized in the dopaminergic terminals of this local module I was telling you about. And one final, I'm almost at the end, one final mechanism, the one that I commented before in one of my, the question I posed before, is that we found out that paraxanthine, the main metabolite of caffeine in humans, has a very strong psychomotor effect in rats. It produces much more motor activation. As you can see here, here you have the effect of caffeine, the effect of theophylline, theobromine, and look at paraxanthine. It was much more locomotor activation. Okay. Also, we found out that this is correlated with a very strong dopamine release in a part of the striatum where caffeine was ineffective in producing increasing dopamine. And we found out that this is related to NO neurotransmission. So we block nitric oxide signaling pathway, then we, with L-NAM in this case, it's called, then what we produce is a counteraction of this effect. To make a long story short about this is because we found that paraxanthine in particular, not caffeine, not theophylline, it has a specific, a selective inhibition of phosphodiesterases, but not the cyclic AMP, phosphodiesterase not cyclic AMP preferring phosphodiesterases, but the cyclic GMP, the ones that are involved in nitric oxide. And we believe that this mechanism can really contribute to probably the motor and reinforcing effects of caffeine. Finally, last slide, to tell you that I've been telling you only about the motor effects and about the reinforcing effects. But we also have the arousal effects. And about that, I didn't have time because it's red now, so to tell you that we have multiple interconnected ascending arousal systems that are modulated by adenosine A1 receptors. And those receptors inhibit tonically this system. That includes the cholinergic system, that includes noradrenergic system, that includes histaminergic system, orexinergic system. Well, caffeine takes the brake off by this A1 receptor-mediated inhibition, and that's the mechanism by which it produces arousal. So conclusions, new concepts. Receptor heteromer and local module facilitates understanding of the functional role of interactions between neurotransmitters in the central nervous system, as well as the mechanisms of central acting drugs such as caffeine. The motor and rewarding effects of caffeine depend on its ability to release the pre- and postsynaptic brakes that adenosine imposes on dopamine neurotransmission by acting on different adenosine A2 and A1 receptor heteromers, localized in different elements of the striatal spine model. And finally, I think it is, no, one more. The arousal effects of caffeine depend on its ability to release the A1 receptor-mediated inhibitory modulation of the highly interconnected multiple ascending arousal systems. And this call that I'm making now, I want to draw your attention to this paraxanthine again, the main metabolite of caffeine in humans. I think we have to study this a little bit more to see if, after all, this compound contributes to the reinforcing motor activating effects of caffeine. And that's it. Thank you.
Thank you, Dr. Ferre. Our next speaker, Dr. Temple, is joining us via WebEx. Dr. Temple, are you there?